We have discussed in the previous lectures about the various aspects of cellular injury, and one of the most important element for the cellular injury is amyloid protein. As you all know that amyloid is a misfolded protein. It's a kind of protein, but due to abnormalities, it is like misfolded, and it deposit in the extracellular space, which we call is ECF. It's known as extracellular fluid or extracellular space. It is not deposited inside the cells, inside the cytoplasm, but it is deposited extracellularly. So, in this context, we can see that it damaged the tissues. It damaged the tissues around the cells. So, one important thing here to worth mention is that multiple proteins can be deposited as amyloid. It means that there are several proteins that share features similar to amyloid, and they can be deposited as well. But the shared features of these are listed below. First is the beta pleated sheet configuration. It means that the proteins that deposited as amyloid should have beta pleated sheet configuration. Second, it should have Congo red staining. It means that under the staining, when we stain that protein, it should be Congo red. And the third is the apple green birefringence. It means that under the polarized light, the protein should impart a apple green birefringence. Two images are shown below, and in the first image you can see that this is the blood vessel, and surrounding the blood vessel we can see a Congo red stain. It's the amyloid, okay, it's amyloid protein. And in the second image, this is the apple green birefringence under the polarized light, seen microscopically. So there are two types of amyloidosis. First is the systemic, and the second is the localized. Systemic means that the amyloid deposited in the systemic like organs. For example, it damaged the blood system, or it damaged the kidney system. But in the localized, the amyloid is deposited in specific organs. So let's talk about the systemic amyloidosis. The systemic amyloidosis is further divided into primary and secondary. The primary means that there is systemic deposition of specific amyloid protein. It's known as AL amyloid. And AL amyloid protein is derived from immunoglobulin light chain. It means that the immunoglobulin light chain, as you know that the immunoglobulin has two heavy chains, and on the sides of the heavy chains the immunoglobulins have the light chain. So this AL amyloid is derived from the light chain of immunoglobulin, and it is deposited in the systemic tissues. For example, in case of plasma cell dyscrasias, the plasma cell dyscrasias refers to the disease of the plasma cells in which the plasma cells are abnormal. For example, in case of multiple myeloma, there is excessive production of beta globulins by the plasma cells. As you know that the plasma cells are nothing but the B lymphocytes. So B lymphocytes mature and they form plasma cells, and these plasma cells start to produce antibodies which we called as immunoglobulins. But in case of the diseases or a set of diseases known as the plasma cell dyscrasias, we can see a disease known as multiple myeloma. Multiple myeloma is a condition in which the plasma cell starts to produce abnormal proteins, and it is deposited as AL amyloid, as we are discussing about primary systemic deposition, right? So AL amyloid is deposited in the systemic type of the amyloidosis. The second is the secondary. Secondary is the systemic deposition of another type of amyloid protein known as AA amyloid. And AA amyloid is derived from serum amyloid protein, or we can say it as serum amyloid associated protein, SAA. SAA holds a very important place in a case of inflammation. SAA is an acute phase reactant. What is acute phase reactant? Acute phase reactant means that those substances or chemicals that are produced in acute inflammation, the first line signals or first line chemicals that are produced in case of inflammation are known as acute phase reactants. And this deposition of AA amyloid is increased in the chronic inflammatory states. It means that when there is chronic inflammation going on, the SAA deposition will be high, and from SAA, AA amyloid will be produced. Okay, it's logical. What are the conditions for the chronic inflammation? First is the autoimmune diseases, for example, rheumatoid arthritis. Second is the lupus, systemic lupus erythematosus. Third is the Crohn's disease, and Crohn's disease is the inflammatory bowel disease, and also other ulcerative colitis and chronic osteomyelitis. These are some chronic inflammatory states in which the SAA is increased, and due to the SAA increased, the serum AA amyloid is deposited, right? The second case in which the acute phase reactant, the serum amyloid protein, is increased in malignancy. In case of malignancy, there is a concept that when there are tumors, there is inflammation going on against that tumors. It means that cells are trying to protect our body from those tumors. So there is inflammation going on between the WBCs and the tumor cells. So in case of malignancy, the third important thing is the familial Mediterranean fever, and it's very unlikely to see in the clinical scenarios, but it's most important in contexts of the exams. But the familial Mediterranean fever means that there is dysfunction of neutrophils. Neutrophils are dysfunctioned. It is due to the autosomal recessive inheritance, you can say, and it occurs in the patients or persons of Mediterranean origin. How will the patient or person presents with the Mediterranean fever? It's the episodes of fever as you would expect, and acute serosal inflammation, again inflammation, right? But serosal surfaces means that, for example, the appendix has its serosal surface, and myocardium has its serosal surface, the our GIT has its serosa. So it can mimic the appendicitis. It means that when there is inflammation of the serosa, it mimics the appendicitis. It can also mimic arthritis. It can also mimic myocardial infarction if there is inflammation of the serosa of the heart. So it is not to be confused with the appendicitis, arthritis, or myocardial infarction, because it is only mimicking them. The actual thing going on with the person is the familial Mediterranean fever. And why is the SAA deposited? Because we have seen that SAA is an acute phase reactant, so its level increased during the attacks, and from the serum amyloid associated protein, AA amyloid is produced, and it is deposited in the system. So we can say that there is secondary systemic deposition is going on. What are some clinical findings of the systemic amyloidosis? So, where we can see the systemic amyloidosis, the most important organ involved is the kidney, and when the kidney is affected, we call it as nephrotic syndrome. It is different from nephritic syndrome. We will discuss the difference between nephrotic and nephritic syndrome in the renal playlist, but here we should mention that in the amyloidosis of the kidney, as the systemic amyloidosis, the kidney will show the clinical symptoms of nephrotic syndrome. Second is the heart. It will cause restrictive cardiomyopathy or arrhythmias. Why restrictive? Because The amyloid is deposited in the walls of the heart, and the walls of the heart become stiff and less compliant. It means that the walls of the heart cannot dilate, right? They cannot fill up with the blood, for example, ventricles and atria. So they are less compliant to the blood due to the deposition of AA amyloid. So there is restricted cardiomyopathy and arrhythmias. Third is the tongue enlargement. Fourth is malabsorption because our GIT walls are deposited with the AA amyloid and there is a malabsorption. Hepatosplenomegaly can also be seen. Diagnostic criteria for the amyloid is biopsy because we cannot just see the serum, we cannot just suspect the amyloid via the blood test, via the CBC, but we should also do biopsy. The most important targets for the biopsy are the abdominal fat pad and the rectum because these are easily accessible for biopsy, and the damaged organ must be transplanted. It's very important clinically because amyloid cannot be removed. It was the systemic amyloidosis, and the next one is the localized amyloidosis. It means that the amyloid is deposited to a single organ. Okay, what are some examples? First is the senile cardiac amyloidosis. As the name suggests that senile, it is occurring in elderly people. Senile means elderly and juvenile means young. So senile cardiac amyloidosis, what it represents? It represents non-mutated serum transthyretin deposited in the heart. Transthyretin is a protein because we are talking that multiple proteins can be deposited as amyloid. So transthyretin is nothing but a protein. It's a serum protein and it can be deposited in the heart. But in case of senile cardiac amyloidosis, it's non-mutated serum transthyretin. The conditions are asymptomatic and present in 25% of individuals over 80 years of age, as we have learned that it's senile. The second is the familial amyloid cardiomyopathy. It is a condition in which the same serum transthyretin will deposit in the heart, but in this condition, the serum transthyretin is mutated. It's mutated. The senile cardiac amyloidosis was asymptomatic, but the familial amyloid cardiomyopathy will be symptomatic. It will show the restricted cardiomyopathy symptoms and it is carried by the African Americans. Okay, the mutated gene, because it's mutated serum transthyretin, so the gene must be carried by someone, so it is carried by the African American. The third is the diabetes type 2. It's non-insulin dependent diabetes mellitus. As you know that non-insulin dependent diabetes mellitus, the pancreas, the islets are still producing the insulin. The beta cells of the pancreas are producing the insulin. The insulin level goes high, and from that insulin a protein is produced known as amylin, and amylin is deposited in these islets of pancreas. Fourth is the Alzheimer's disease. And Alzheimer's disease, it's worth mentioning that uh, the some proteins are deposited in the brain, forming amyloid plaques. What are those proteins? These are A beta amyloid. A beta amyloid is deposited in the brain in case of Alzheimer's disease, and this A beta amyloid is derived from beta amyloid precursor protein. Why it's worth mentioning that beta amyloid precursor protein? It is because that the gene for beta amyloid precursor protein is present on the chromosome number 21. Recall from your secondary level biology that the 21 chromosome trisomy is known as the Down syndrome. It's trisomy of 21. When there is the Down syndrome, there is a trisomy of the 21 chromosome. The gene for the beta amyloid precursor protein will be activated earlier, It means that those people with Down syndrome will develop the Alzheimer's disease by the age of 40. It's the early onset. They would develop the disease earlier than would expected. Okay, so this was for the Alzheimer's disease. The last but not the least is the dialysis associated amyloidosis. What is dialysis associated amyloidosis? It means that the beta 2 microglobulin will be deposited in the joints. But why in the joints? Let's see the context, and then we will understand that why beta 2 microglobulin, it's a protein which will represent as amyloid, will be deposited in the joint. First thing, that there are MHC class 1 molecules that are present on the surface of many cells. It has transmembrane domain, this one, and it has an extracellular domain, right? You can see here it's MHC class 1 molecule. It has a transmembrane domain and an extracellular domain. But the supportive structure for the MHC class 1 molecule is beta 2 microglobulin. Okay, it's beta 2 microglobulin which provides the structural support to the MHC class 1 molecules. When there is dialysis going on in case of the renal failure, this beta 2 microglobulin is not filtered from the blood because the kidney is not functioning and the patient or the person is on dialysis. When the beta 2 microglobulin is not filtered, its level in the blood will increase and it will deposit it in the joints as amyloid. So this was the history or the context of the dialysis associated amyloidosis. The last is the medullary carcinoma of thyroid. Medullary carcinoma of thyroid, in which there are tumor cells and that will produce calcitonin because the tumor cell consists of C cell. It's a C cell tumor. The C cell tumor will produce calcitonin and calcitonin will deposit it within the tumor. And it's very important to mention that microscopically we can see the tumor cells in an amyloid background. It means that there are a lot of amyloid. It means the calcitonin. Calcitonin will be a lot of in the background and the tumor cells are present, right? These are tumor cells and the calcitonin or amyloid will be in the background. So this is the microscopical representation of the medullary carcinoma of the thyroid. Okay, so this was for amyloidosis. I hope you understand this video and I will see you in the next video. Till then, goodbye.